to our April series of the Black and Neuro Seminar Series. We are so excited to have Dr. Termara Parker here. As we start, I wanted to point out that automatic closed captioning is available. You can access this by clicking the three dots in the bottom right corner of your screen. And if you experience any issues accessing these captions or interpreters, please message in the chat to the panelists and hosts. I'm Casey Dieters. I am a assistant professor at UC Los Angeles, and I am the Black and Neuro Seminar Series program lead. And I am, again, very happy to welcome everyone to our seminar series. In this series, we plan to feature neuroscience faculty, neurologists, postdocs, and graduate students. We hope to provide a stage for them to share their science, their paths, their setbacks, and their triumphs. And we hope through this, we can celebrate our community and help change the broader community's ideas of what a neuroscientist or neurologist looks like. Some logistics. After I introduce Dr. Parker, after the talk, we will have about 10 to 15 minutes for questions from the audience, and you can ask them using the Zoom's Q&A function at the bottom of your screen. Feel free to share those throughout the talk, and I'll moderate them again at the end. Additionally, if you have any tidbits you want to share from today's seminar on social media, please do so using the hashtag BIN Seminar Series. So with that, it is my honor again to present today's speaker, Dr. Termara Parker. Dr. Parker recently defended her PhD in the Interdepartmental Neuroscience program at Yale University. While at Yale, she studied the neural mechanisms of social interaction in autistic individuals using functional near-infrared spectroscopy and eye tracking. Before attending Yale, she graduated summa cum laude and Phi Beta Kappa with a bachelor's in biology from Howard University. Dr. Parker has authored 13 publications, which includes 7 first author publications, has won 3 prestigious awards, such as the NSF GRFP, the Neuroscience Scholar Program Fellowship, and the National, I can't say this word, Bouchet Graduate Honor Society?
Bouchet.
Thank you. Yes, Bouchet Graduate Honor Society during her time at Yale. In addition to her passion for science, Dr. Parker's commitment to outreach strengthened her desire to teach and mentor future Black neuroscientists. Yes. In 2020, Dr. Parker was named the graduate director at the Yale BBS Diversity and Inclusion Collective. As a graduate director, Dr. Parker has been instrumental in the creation of resources and opportunities for underrepresented minorities to build community through the biological and biomedical sciences. Wow, so impressive. You definitely earned that PhD, and thank you so much for all the work that you've done mentoring other Black neuroscientists. And so with that, I'm going to turn it over to you, Dr. Parker, and I'm very excited to hear your what you have to tell us. Thank you.
Awesome. Thank you for the introduction, Dr. Dieters. So I'm going to share my screen now. Okay. Everybody can see my screen? So I just want to start off talking about what my why is before I start talking about my research, because I think this is important for trainees and students who may be in the lab right now who are kind of like and the loss of words and trying to figure out, okay, my experiment's not working, but what's the reason I'm doing this research? So I just want to talk especially about what is my why, what got me into science. So first, I am the oldest daughter of 4 daughters. I grew up in this small town in Bedford, Massachusetts, in which I was one of the few Black people in my town. And in which one of my sisters was diagnosed with autism spectrum disorder. And she was diagnosed with the disorder at the age of 9. And I had always been curious about how did my sister really understand the world around her. And I knew that my family, unfortunately, we had not really any clue on like, how do you really help treat and understand somebody who's autistic? So this led me to be very curious in high school, in which I did this program at Harvard Medical School every Saturday between December and February, in which I was exposed to a plethora of health professions, and I was able to be involved in health research. And I had a mentor who actually came to my thesis seminar, because I actually told her about this, and I was able to study autism spectrum disorder, and basically focused on the biomedical interventions with autism spectrum disorder. And it was very interesting because my mentor also had an autistic sibling. So we both were able to really commemorate in those fashions. And that also helped me understand, like, I'm actually very interested in understanding what it's like to understand somebody who's autistic. What can we do to help make things better for them? And it just helped open a whole bunch of doors for me, because I realized, like, oh, it's not just about science, it's not just about blood. I can actually do some research on it. At the time, I didn't know too much about the PhD process. So I decided, hey, I want to go pre-med track. I want to be a neurologist. So I ended up enrolling at Howard University in Washington, D.C., and I got my bachelor's degree in biology, in which I did some research focusing on simian immunodeficiency virus, which is SIV, which is very similar to what you would see in HIV in humans, but it's specific to macaque monkeys. And I was interested in how the virus affects the brain, particularly looking at the hippocampus and amygdala. I also did some research at MIT, in which I was focusing specifically on what my biggest passion was understanding autism spectrum disorder, and look at the neural mechanisms of face processing using a mobile eye tracker, as well as using fMRI. Then after I did my summer research programs and finished at Howard University, I enrolled at Yale University, in which I started my PhD in the interdepartmental neuroscience program. And I was there for 5.5 years, and I just defended my thesis November 29th. So I'm just happy to just talk about, I just want to first talk about what is my why, because I think it's really important as scientists to really talk about what gets us really into science, because that's what causes us to be the scientists we are, because we are passionate about, like, helping the world. So going into my topic, some of the topic of my thesis work was entitled The Neuroprocessing of Eye Gaze and Autism Spectrum Disorder. And I did this in the Brain Function Laboratory under the direct mentorship of Dr. Joy Hirsch. So first, I'll be going into the background of autism spectrum disorder, or ASD. Then I'll be going into the methods. I'll be focusing on functional near-infrared spectroscopy. I know it's a mouthful, but we'll talk about that. Then I'll talk about this eye gaze phenomenon known as joint attention, go into some discussion, limitations, and future directions, as well as acknowledgements. So firstly, talking into the background of ASD, I will be talking about the behavioral and neural evidence of atypical processing of eye gaze in this disorder. So autism spectrum disorder is defined as a neurodevelopmental disorder that can be characterized by difficulties in social communication and social interaction, and restrictive and repetitive behaviors and interests. And these ranges can manifest in different unique behavioral symptoms, ranging from severe to even just mild, and depending on what the individual is. For example, we can look at this schematic. We can look at a female autistic female might be relying heavily on other children to guide and speak for her, maybe often described as quiet or shy, or perhaps have epileptic seizures. So to overcome this complex heterogeneity of ASD, you must identify the underlying processes that govern social behavior and determine whether these systems are disrupted in autistic individuals. Something important to note is that as of at the end of last month in March, 1 in 36 children are autistic. When I first did my thesis seminar, it was actually 1 in 44. So it has increased over time. And because of this, this has made a lot of researchers been interested in understanding the pathophysiological markers specific to autism spectrum disorder. So to really understand these building blocks of autism spectrum disorder, or ASD, researchers have really focused on ASD in all different levels, from the genetics level to the systems level. And despite all of this effort, unfortunately, we still haven't had a clear biomarker for ASD. And this may be due to many reasons. One reason that we can think of is that ASD, as I talked about, is a social disorder, and it's really important for us to really include the social context when we're doing the research practices that we're doing. And one social symptom that we typically see in ASD is atypical gaze patterns and avoidance of eye contact. These have been replicable diagnostic features. So it's really important for us to really include these type of symptoms into our research practices to really help uncover some of the possible biomarkers we could see in ASD. Previous research has shown two potential explanations for the aberrant processing of information from the eyes. Number one is that there's this general lack of social attention, which results in the missing orientation towards social cues from the eyes. And then two, that there's this aversion of eye contact leading to the avoidance of eye fixation. And these two explanations were proposed by Spezio and colleagues in 2007. And Spezio pointed out that these explanations do not have to be mutually exclusive. While many studies have investigated these theories regarding eye contact and ASD, the exact associations between the ocular motor responses, the eye responses, and the neural signals necessary to gather salient information from the eyes still remain unclear. So to kind of focus more on this atypical processing of the eyes, we're going to first ask this question. What is the behavioral evidence for atypical face processing in ASD? So first, Pelfrey and colleagues in 2002, in the seminal paper that was cited over 1,000 times, focuses on 5 male autistic individuals and 5 typically developed or controlled participants as they looked at emotional faces. And what was found is that the autistic individuals tended to look at external regions of the face as opposed to typical developed individuals that tend to look at the core regions of the face, such as the eyes, nose, and mouth, which showed that there are abnormal fixation strategies that are seen in autistic participants, and that they fixate less on the core regions of the face as opposed to they were focusing more on the external regions of the face. Similarly, we see in Dalton 2005 paper, again with emotional faces, which also include quarter-turned pictures of faces. We saw that typically developed individuals tend to look at the face longer than the autistic individuals. And also when it came to the eyes and the mouth, it seemed that the typically developed individuals tend to look at the eyes more versus with the autistic individuals, they didn't have preferential fixation between the eyes and the mouth. So again, showing these abnormal fixation strategies that we see behaviorally when it comes to looking at faces. But the next question is how do these abnormal fixation strategies actually map onto the brain? So the question is, what are the neural correlates of face processing in ASD? So there are many techniques that we can use to study the neural correlates of face processing, but I'm going to first focus on something that has been used time and time again, which is the electroencephalography, or EEG, which can be used to detect the initial stages of face processing, and which you can have a net on your head, some electrodes on your head while you are engaging in behavioral tasks. So electroencephalography, or EEG, is thought to result primarily from the synchronization of postsynaptic potentials at cortical pyramidal neurons. And the recorded EEG signal does not represent single neuron depolarization as opposed to a plethora of neurons. And you can have this negative depolarization, or you can have this positive depolarization that occurs. So a negative depolarization will give you the N versus a positive depolarization will give you a P. So when we look into Benton in 1996, when he was focusing on this particular negative depolarization that was seen in these posterior areas of the brain, he saw in typically developed individuals that there is this robust activity that we see in T5 and T6, was kind of in the posterior parts of your scalp, and saw that this activity seems to happen approximately 170 milliseconds after a stimulus presentation, and seems to be specific to facial features and even strongest to the eyes. And this was known as the N170, which is a negative peak over the lateral posterior regions of the scalp, approximately 170 milliseconds after stimulus presentation. So using this particular component, we see this is specific in typically developed individuals. If we see there's abnormal fixation strategies, then we would hypothesize that perhaps that we'll see an abnormal activity of the N170 in autistic individuals. So when we look at McPartlin et al. 2004, and we use these, the posterior regions of the scalp again to really focus on this N170, we focus on children and adolescents who looked at pictures of faces, inverted faces, and furniture. And what was found is that the TIPI developed individuals seem to have this activity of the N170 that we showed in the last slide, and is very strongest for faces. And then when we look at the ASD group, they seem to have this slowed N170, which is not as fast as the TIPI developed group. So we show that there's a slowed neural speed of face processing in ASD. So this was seen in McPartlin, which was my first thesis advisor's lab. And then this was also replicated that this N170 could be a potential marker in the Kang 2018 paper. But the next question we have is like, okay, we see that there's differences in the face, but where on the face are we going to see these neural differences? So my first author paper, I focused on this question: is slow neural speed of face processing dependent on where participants look at the face? So if you look at the eyes, nose, or mouth, where will we see differences in this neural activity? And I was specifically focusing on children and adolescents with or without autism. So the N170 was seen to be most delayed with the eye. So the N170 delay is more pronounced when the attention is directed to the eye region of the face, as opposed to the nose and mouth, which is providing evidence for the lack of social attention theory. And this key finding can elicit further investigation of disrupted neurobiological pathways specific to eye gaze and early stage face processing. So to summarize the first part, what is the behavioral evidence for atypical face processing in ASD? So we see that autistic individuals fixate on external regions of the face, and that autistic individuals do not preferentially look at eyes over other facial features. And what are the neural correlates of face processing in ASD, was we asked. And we saw that the N170 latency to face is delayed in ASD, and that the N170 latency is most delayed when attention is directed to the eyes. But as I talked about in the beginning, we need to understand autism in the most social context that we can. So we're talking about passive looking of faces, but we know that in terms of social interaction, looking at a picture of the face is not the same as actually looking at your friend while you are just hanging out in the bar. So if we go even further, what are the neurobiological pathways specific to eye gaze under naturalistic settings in ASD? So if we look at this image from Blackish, for those who watch Blackish, eye gaze can play a significant role in live social interactions by directing the acquisition of facial cues. In face-to-face interactions of typically developed individuals, both adults and children focus their attention preferably and reflexively towards the direction of another person's gaze. And despite the social significance of known typical responses to eye gaze, the underlying neural processes of live eye-to-eye contact are not well understood. And this knowledge gap may be due to limitations of conventional neuroimaging methods that fail to capture natural two-person interactions. Our lab studies natural two-person interaction using FNIRS technology, and with this novel ability to study live two-person interactions, investigating of typical and atypical responses to real-life processing of eye gaze are possible using simultaneous eye tracking and FNIRS, or functional near-infrared spectroscopy. So I'm going to go into talking more about functional near-infrared spectroscopy, also known as FNIRS. So FNIRS is an optical imaging method that measures changes in light scattering and absorption properties of the tissue following neuroactivity. And these hemodynamics are acquired from superficial cortex by FNIRS, and it distinguishes variations in the oxyhemoglobin and deoxyhemoglobin signal. So we kind of think of this in action. So first we will have something that would be called an emitter versus a detector. So those are one of the options, could be emitter or detector. And then the emitter actually transmits 3 rays of light that can be absorbed by the deoxyhemoglobin or oxyhemoglobin or both. And then the detector is able to detect what light was not absorbed by the cortical tissue. So as you can imagine, you can only get activity from the cortical areas of the brain as opposed to the subcortical area. So that's one limitation that we might have on FNIRS. But if we think about a particular task, like the finger thumb tapping task, in which you'd be going like this for 15 seconds versus 15 seconds of rest, and we're focused on a channel in the motor cortex, we'll see oxyhemoglobin will go up during tap, down during rest, up during tap, down during rest. And then it will go the opposite, deoxyhemoglobin will go down during tap, up during rest, down during tap, up during rest. So these two hemodynamic signals are acquired using this FNIRS technology. And in typical FNIRS analysis, the FNIRS signal is commonly regressed via a general linear model, which is constructed by convolving the hemodynamic response with the experimental task. And this analysis can be used with combined hemoglobin signals. So you can have the sum of the oxyhemoglobin and the inverted deoxyhemoglobin, which is what we use in our lab. And to remove these physiological non-neural components, we can use a principal components analysis to get the signal that we would like to be able to do these results. For example, doing condition A greater than condition B, or A greater than condition A greater than rest. And when we think about the advantages of FNIRS, it replicates the blood oxygenated level dependent signals acquired by fMRI. And that is tolerance of limited head movements. So this can be helpful for those in clinical populations. The temporal resolution is 30 milliseconds, which is much faster compared to the fMRI, which tends to get signal every 2 seconds. The spatial resolution is 3 centimeters. As I said, you won't be able to get kind of the subcortical areas, but you'll get some high functioning cortical areas. So you can have simultaneous acquisitions of multiple modalities. So you could do skin conductance, eye tracking while doing FNIRS. And lastly, I think what's really helpful about FNIRS is that it allows the assessment of neural responses in a broader range of clinical participants who may not be able to go into an fMRI scanner because of claustrophobia or the fact that you have to stay super still. So in our lab, we were able to use FNIRS to study the neural correlates of live eye contact in ASD, in which we had a participant, whether it be an autistic participant or a TB developed participant, and a lab partner, which engaged in this 15-second eye contact and 15 seconds of rest. And we did this while acquiring eye tracking as well. And this paper was just published the end of last year. And then in the results, we saw that typical developed participants with eye tracking, we see this robust activity in the dorsal parietal area, as well as the dorsolateral prefrontal cortex. And this is when we're comparing live eye gaze greater than rest. And when we look at the ASD participants, we don't see this activity in this dorsal parietal area, which includes the somatosensory association cortex and supramarginal gyrus. But instead, we see this alternative circuitry that we see kind of more ventrally in the brain. So when we go back to the question we had in terms of understanding the neurobiological pathways specific to eye gaze under naturalistic settings in ASD, we see that the comparisons between ASD and TD showed there's this decreased dorsal parietal activity in regards to live eye contact, and increased ventral temporal parietal activity for ASD during this live eye contact condition. But just to even test further, we also know that just staring at each other for 15 seconds is not going to really give us what it really is in terms of like how it is in social interaction settings. So in my research, I focused on this phenomenon known as joint attention, which I'll talk about in the next slide, which is focusing more on the dynamics of the eyes. When your eyes looking one way versus the other way, how do you interpret your partner's eyes as they look to the left or the right? So typically developed individuals naturally focus our attention towards the direction of another person's gaze. And this two-way exchange of social information is defined as gaze cueing of attention. And gaze cueing of attention facilitates joint attention, which involves the sharing of attention between two individuals, as we see right here, towards the same object. So Sally could be looking at the butterfly, and then that's when Joe will say, oh, I'm seeing that Sally's looking at the butterfly. I'm going to reflexively look my eyes at this butterfly. So this is an example of nonverbal communication. And engaging in joint attention is vital for developing social, cognitive, and communication skills throughout life. And this may be a potential area of interest for understanding joint attention difficulties in ASD. And joint attention can only happen due to visual sensing. And visual sensing involves the active sampling of relevant social information using a systematic pattern of eye movements and fixation. So our eyes and our brain uses this visual sensing mechanism to know, okay, I need to quickly look at that direction of the room because I'm seeing everybody looking at the left hand. This can be very helpful if you see threats or maybe just something that's very intriguing. So in our lab, we have seen that there's specialized neurocircuitry for real life eye gaze and joint attention. As I showed a couple of slides ago, we showed that the dorsal parietal area seems to be specific to live eye contact in typically developed individuals. Similarly, Swetha Dravida in 2020 used this task, this is known as the joint attention task, in which this is the view of the participant, in which the participant will be looking at a partner, and the partner will be looking at 3 dots on this particular glass experimental apparatus. And then in another condition, the LED light will indicate to either look at the blue, green, or red. So that will indicate social joint attention, means the partner will be using their eyes to show you where to look, versus the LED display would be a non-social joint attention condition. So when we compare social joint attention and non-social joint attention, we see that the right temporal junction seems to be most active during the social joint attention condition, and this was seen in typically developed adults. And this particular result has been similar to previous results in the lab, which have shown this activity that seems to be specific to live eye contact. So we are again showing, okay, it seems like this right temporal parietal junction area and this dorsal parietal area seems to be specific to live eye contact. And moreover, the right temporal parietal lobe is modulated by the frequency of eye contact. So when you separate low eye contact pairs, so those who are not looking at their partner's eyes as much versus partners who look at the partner's eyes a lot, you'll see that the right temporal parietal junction is positively correlated with eye contact events. So the more you look at your partner's eyes, the more activity you'll see in this particular area. So if we're using this, we have a lot of questions. Okay, so if we're trying to focus on joint attention in ASD, what questions can we ask, and what can we essentially hypothesize? We tried to do this task with autistic individuals, but unfortunately, we were unable to be successful in making sure that the autistic individual was able to engage with the lab partner. So we tried to make the task a bit simpler and just try to really find ways to really separate the gaze cueing part of the joint attention phenomenon. So the first question we have is, what are the neural and visual processing mechanisms involved in live joint attention in ASD? And I hypothesize that visual sensing mechanisms will be altered and associated with dorsal parietal hypoactivity in response to live human gaze-induced joint attention. And this is because of what we've seen in the previous research. The second question, I was very interested in really linking brain and behavior. How does the neural processing of live joint attention relate to behavior? How does it relate to these autistic traits or these standardized assessments we use to study autism? And I hypothesized that dorsal parietal hypoactivity during live human gaze-induced joint attention will correlate with symptom severity. So to introduce my paradigm, we have this smart glass that was in between the initiator, which is this lab partner, and the participant, which could be either typically developed individual or an autistic individual. We used this smart glass. It wasn't because of the pandemic, although it was also very perfect during the pandemic, since that's when I was doing a lot of my data collection. But it electronically is controlled to go opaque during the rest and transparent during the task periods. So how it goes is that if you are a participant, you'll look 3 seconds at the smart glass, focus on the fixation cross in the middle, and then the smart glass will open up and you will see your partner, and your partner will make eye contact eye contact with you for 2 seconds, and then we'll look at the left or right green dot that's located on the smart glass. And your job will be to follow the eyes of the human initiator. And I call this condition the human gaze cueing condition. I also did another condition, which is very interesting, and I think it's the one that's like everybody gets very like intrigued about, is that I want to see, okay, if we're using joint attention and eye movements, we want to make sure this is specific to human eyes and not just any type of dynamic eyes that we can see in cartoons. And we know that autistic individuals in many research studies have shown that they're really better at engaging in joint attention with robots. So I was interested in using this same type of paradigm I showed previously, but in a robot to see, okay, are we going to see differences in just any type of joint attention, or is it specific to human eyes? So I use this robot, which is what we call it Maki. And then the Maki robot does the same thing as the human partner, and it engages in looking at you for 2 seconds, and then it's going to look at the left or right green dot that's on the smart glass. So same experimental paradigm that I showed, but instead of the human, it's a robot. And I call this the robot gaze cueing condition. And the task was the participant follows the eyes of the robot initiator. And in terms of the experimental timeline series, we have the eye to eye contact is first 2 seconds, and then green is the gaze direction left or right, and then black will be the resting period. So I have 5 blocks, and I had 3 trials per block, and then 180 seconds per run. I did 2 runs for gaze cueing, human gaze cueing, and 2 runs for robot gaze cueing. In terms of the methods, I use eye tracking, and I also use FNIRS, and I did full coverage of the head, both hemispheres, 40 emitters and 40 detectors. And I did standard assessment, and I used the BOSA, which is the brief observation of symptoms of autism training, which was developed during the pandemic in a way to study autistic traits in a way that through telehealth or with PPE equipment. So it's very similar to the ADOS, or the Autism Diagnostic Observation Schedule. In terms of participant demographics, I had 30 typically developed individuals and 20 ASD individuals that were in their mid-20s. And because I said when we did the same experiment that Swetha Dravida created, and unfortunately the autistic individuals weren't able to complete the task, we wanted to make sure with this task, were we going to get some good compliance? And we were able to successfully do this task in which the individuals were able to gather information from the face and look at the left and right green dot on the smart glass. And because I'm very interested in this kind of visual sensing process and how do we gather information from the face to really engage in gaze cueing and joint attention, I was interested in this visual sensing component known as dwell time. And dwell time is the number of valid retained samples per interval normalized by sampling rate. And I'm focusing on the first 2 seconds because that's when you're gathering information in the face before the movement occurs. So we can relate socialness of gaze cueing as measured by the average duration spent on the human face before making the appropriate eye movement. So although I did not see significant differences between the ASD group and the TED group with the human gaze cueing condition, we did see differences in the robot condition, in that the ASD group tended to have significant differences in looking at the robot and looking less at the robot compared to typically developed group. And this may be perhaps because the ASD group might say, okay, the robot is very predictable versus the human partner is actually a little bit more dynamic and not as predictable. Because when you have a robot's gonna go like very stereotype behavior. And then even though I didn't see differences in how they looked at the face of the human, we do see a little bit of a trend. And I also wanted to see how does this map on the brain? Maybe they're looking at it around the same rate, but maybe it's mapped on a different area than we would predict. So first, I use this dwell time as a modulator of the neural signal. So In the red, we can kind of see these are the individual dwell time markers. So you could have been looking at the face for 1.5 seconds versus 1.3 versus 1.3 seconds. And then you can change all through the entire run. As I said, there's 3 trials per block and 5 blocks per run. So I use the individual dwell time and correlated that with the hemodynamic signal to create this signal that is dependent on how long you're looking at the face, which kind of helps us map this visual sensing acquisitions and the hemodynamic signal. So what I found is that human gaze cueing greater than rest with the dwell time modulation show this dorsal parietal activity that is very similar to what we showed like a few slides ago when we were looking at live eye contact. But the ASD group did not show this activity in the dorsal parietal area, but instead show more activity in the dorsolateral prefrontal cortex. So again, showing hypoactivity in the dorsal parietal area, and then also alternative circuitry in the frontal area. When we compare this with the robot, we see that this dorsal parietal area seems to be really specific to human gaze cueing and not necessarily any type of gaze cueing. So again, kind of really validating that the dorsal parietal area is specific to human social interaction, because in the TD group, we see more kind of supramarginal gyrus, angular gyrus, but not so much somatosensory association cortex. The ASD group was interestingly had some dorsal parietal activities such as the somatosensory association cortex, which is kind of showing, okay, it seems like the ASD group might be trying to recruit some of the dorsal parietal area for the robot, but instead of the human, which again is even emphasizing that there is a more alternative circuit that is maybe indicating that perhaps like they might see the robot a bit differently than the typically developed individual. So the second question I had, since we're seeing these differences, how does neural processing of social gaze cueing relate to behavior? How does brain relate to behavior? So reductions in dorsal parietal activity is what I hypothesize is that reductions in dorsal parietal activity during human gaze cueing will relate to symptom severity. So as I talked about, I used a brief observation of symptoms of autism training. And with this particular standardized assessment, if you have high BOSIS scores, you would have moderate to severe ranges of autism. If you had low BOSIS scores, you would have little to no or very, or even like mild ASD. So I use this to correlate that with the hemodynamic signal. So the blue clusters indicate that higher BOSIS scores are associated with reduced beta values to real face gaze. So that when you have increased autistic symptom severity, that it indicates decreased right dorsal parietal activity. So kind of in more simplistic terms, if you are a participant who had a BOSIS score that was on the mild range, and you would probably have a higher activity in that somatosensory association cortex and supramarginal gyrus area. But if you were somebody who had more severe autistic traits, you might have lower activity in that particular area. So it was actually a very strong correlation. It was R negative 0.86. So it was very interesting to see that this was such a strong correlation between these autistic traits and the hemodynamic signal. And when we look at the robot doesn't have this negative association, and that actually when you have the individual symptom severity positively relates to right dorsal stream activity. So this right dorsal stream activity isn't disrupted by the autistic traits. They seem to be able to recruit this area when it came to robot gaze cueing greater than rest. So taking this all together, what have we learned? What are the neural and visual processing mechanisms involved in live joint attention in ASD? We learned that oculomotor mechanisms drive reduced dorsal stream activity and increase alternative frontal processing in response to human gaze cueing, but not at robot gaze cueing. And how does neural processing of live joint attention relate to behavior? And we see that individual autistic symptom severity relates to reduced right dorsal parietal stream activity during human gaze cueing, but not robot gaze cueing. So if we just take a second to kind of talk about all we've discussed so far, because I know I've given a lot of results, but how do we take all of this results and synthesize what we've learned? So first, we saw initially that autistic individuals tended to fixate on external regions of the face and do not preferentially look at eyes over other facial features behaviorally. We also saw that when we use EEG, we can focus on this N170 component that's specific to the neural correlates of face processing. And we see that the N170 is slowed in ASD, and particularly this slowed neuroactivity occurs when the autistic individual is looking at the eyes as compared to the nose and the mouth. And then when we focus on more naturalistic context, we see that the neurocorrelates of live eye contact in ASD, we see that there's alternative circuitry at play in which we see ventral activity in the ASD brain versus more dorsal activity in the TD brain, in which that they saw hypoactivity of this somatosensory association cortex and supramarginal gyrus, which is known as the dorsal parietal area. So the dorsal parietal area seems to be hypoactive in the ASD brain. And then when we go a step further and focus on joint attention, which involves information gathering as opposed to passive eye contact, we see again this activity in the dorsal parietal area in the TD participants, but we don't see it in the ASD participants. And we again see some alternative circuitry at play. And then lastly, we see this possible biomarker for ASD in that it seems like this dorsal parietal area seems to be correlated with the BOSA covariate, which BOSA can be seen as this autism symptom severity. So dorsal activity seems to be related to BOSA scores. And when we talked about the two potential explanations for atypical processing of the eyes, I introduced the general lack of social attention and aversion of eye contact. And I've shown a lot of evidence so far of the general lack of social attention, in that it's not that they're looking at the eyes less, but however, it seems like the eyes are not as salient, the human eyes specifically are not as salient in the autistic brain as opposed to typically developed brain. So to go into some limitations, as I said before, we're unable to capture subcortical neural responses in using FNIRS. And also, we also see that the pool of participants are not racially and ethnically diverse. So I want to focus on this first limitation and what we can do in the future to help make research more inclusive, especially neuroimaging research. So if you notice from my research, I only had 2 Black TD participants and I had no Black autistic participants. And this is not something that's specific to just my lab. This is a problem that we see in all of human neuroscience research, in that there's this overrepresentation of white participants and underrepresentation of all other minority groups. And this paper we just published in January of this year. We ended up making the cover of Nature Neuroscience, in which we were focusing on the exclusion in human neuroimaging methods and how we can move forward from there. So this is I love using this particular picture because it really shows kind of this systematic exclusion that we see, that we might see white participants come in for fMRI, FNIRS, and EEG research, but then the people who are not included in the research tend to be the Black and brown participants, some who might have hijab, some might have braids or locks. And that shows that there is problems in the research practices that we're doing, and it has to change in order for us to really help make things better for all people. We can't just be focusing on just the white male autistic individuals and not anybody else. So the question that I lastly want to talk about is, how do current recruitment and methodological efforts impact the equity and generalizability of scientific discoveries across the global population? So this paper, which I was working with Teresa Girolamo about, we talked about convenience sampling. And convenience sampling happens is that you can be very well-intended, and you could bring in a diversity of participants from the community. But then when it comes to recruitment, you end up having this convenience sampling that might occur when you are screening the participants, you're doing consenting, or any of those things, or even just putting the FNIRS cap on your head, you're going to start excluding people because, for example, somebody might have intellectual disability and they're excluded from the study. Or it could be because somebody is bilingual or somebody who's maybe have limited English and unable to really understand the consent form. And then thirdly, if you are coming in for FNIRS or EEG research, and then you come in with locks or braids, you may be excluded from the study, or you might actually be able to do the study, but you're excluded later because you're not considered a good participant. So we end up having this homogeneous sampling of participants. And then even further, we end up having underreporting that occurs in that people will not put in their papers why some participants were missing. Is it because of skin type? Is it because of hair type? And not really addressing, oh, we excluded participants, but we're actively looking to try to make things better. So this is something that tends to happen in a lot of human neuroimaging research, and it's something that needs to definitely be addressed. So just to even put it in more simpler terms, convenience sampling, you could have somebody coming in with braids and they'll be excluded from the study. Somebody coming in with an afro, they might be able to do the task, but then there's poor signal quality. If you're somebody with locks, you probably will be excluded from the study. If you're somebody with a wig, you're probably gonna be excluded from the study, or you might have this uncomfortable situation where a lab member might ask you to take the wig off. So this kind of really just shows like, this is not just like a inconvenience problem, this is a systematic problem, and it's definitely affecting more groups than another group. I don't have to point it out, but you can see it. So the next question is, what are the possible solutions? So what are possible solutions that we can do? So first, we can improve science communication. So researchers should actively seek to communicate the importance of science to underrepresented communities and advance public understanding. We can remove financial barriers. So researchers should reconsider the payment method and compensation for study participation. And the core facilities and reimbursement schedules are often designed for university undergraduates, as we know, and not for the general population. So we need to consider accessibility of participants, maybe help with time travel spent traveling, distance from the mass transit, cab vouchers, childcare, help with all those different factors. Also form community partnerships. So researchers should recruit beyond the boundaries of academic institutions. Ideally, inclusive sampling should include partnerships between the patient and the public and researchers. And this needs to be active participation, like whether that be you going to the community and not having the community always come to these ivory towers. And then also focusing on demographic collection and reporting. As I said, there was an underreporting of what's happening when it comes to data collection. For example, in 2008 through 2009, less than 1% of human cognitive neuroscience research articles actually talked about race and ethnicity or socioeconomic status in the research. So we need to be better at actually saying, explicitly reporting the participant demographics in publications and acknowledging the regulatory differences that may constrain the collection and dissemination of demographic data across countries. And then also, lastly, although this is not, there's so many other solutions we could keep coming up with, but kind of one of the big thing is to remove the methodological barriers. And I just kind of want to shout out Arnell Etienne, as well as Jasmine Kwasa, who have been working tirelessly on helping creating EEG electrodes that are suited for individuals with coarse and curly hair. So we need to continue to provide culturally appropriate study materials. For instance, MRI safe hijabs, turbans, and bonnets. And we also need to just also with MRI machines, allow for the recruitment of participants that are over 250 pounds, because it depends on the board that you use. If you use a general standard board, the limit is less than 250, and that can also exclude people as well. So we need to be able to exclude these methodological barriers in order for us to really have research that's more inclusive. And I just want to end with this quote, which I've already said many times in I did in my thesis seminar. I also did it a couple of times in some of these media articles, but I'm gonna say it again, that science cannot help society if it does not reflect society. We cannot keep doing research if it's not reflecting the general population. So these solutions that I have just talked about, we need to really keep in mind. And we also cannot just rely on our Black researchers to help change the systemic problem. This includes everybody. Like, everybody needs to collectively work on trying to help this problem. And I just would like to acknowledge the Brain Function Laboratory, my PI, Dr. Joy Hurst, research faculty, research assistants, graduate students, program managers, visiting fellows, and some of our UCL colleagues, as well as the McPartland Lab, who was helped with ASD recruitment. My program, INP, funding, and then my thesis committee. And I just would like to say that April is Autism Awareness Month, so use this month to help advocate for more understanding of autistic individuals. Also listen to autistic people when they're talking about the research practices that can be problematic. So definitely remember to just keep listening and not necessarily just do science and not really pay attention to what the general public is asking. And also, if we just kind of just think, I just want to just talk about my sister, because I talked about that in the beginning. This is the reason why, like, I'm so passionate on trying to make this research even better, is because how can I help someone like my sister who's a Black autistic individual if she's not included in the studies? So with that, I just would like to end it, and I look forward to any questions from anybody in the audience. Thank you.
We're going to clap, Dr. Parker. You really had me at the, I don't know if you could see on the screen when you were presenting, but you have had consistent hearts, claps, smiley faces, thumbs up, whatever that emoji is with the confetti and the mega horn. I really, really, really appreciate this talk you gave, not only talking about your experience, sharing personal stories with us, but then also advocating for our community. We definitely need more people like you in this world. And I just want to say thank you so very much for like literally all that work, wonderful work you've done. And I can't wait to see what you do with your future. We do have about 6 minutes left. And so we have 3 questions in the Q&A. So we'll start with that. Okay. First, we have a question from Cassidy Willie Laws. Would you be able to repeat the specific method that you used in terms of the seconds of eye gaze?
Oh, yes. So in terms of the eye tracking, I have it so that it was 2 seconds of looking at the participant, and then 2 seconds of when they're looking at the 2 dots that are on the smart glass. And the reason I separated that because I want to see the periods of gathering information from the face versus the periods of when the eyes move.
Excellent, thank you. Next question from Maria Bennett. Does FNIRS activity change in ASD people when looking at higher value people like family or friends?
That's a very interesting question. So we didn't study that because these were definitely just randomized people that came in, but there has been some studies in the literature that have shown that if you are looking at eyes of a familiar family member like your mother or your father, you tend to have a higher, for example, if you look at EEG, higher N170. For FNIRS, you do seem to have more activity in kind of this occipital temporal area, which is very similar to the FFA or fusiform face area. So that's a really interesting question. We weren't able to do it with our small sample size, but I think it's something that will be good for the future.
Excellent, thank you. We have another question. Thank you for this presentation. Can you go a little deeper on the results between ASD patients in a human eye-to-eye contact test? I would have hypothesized that there would be a significant difference, less eye-to-eye contact behavior for the ASD patients.
Yeah, that is something that we also were thinking too. We were thinking at first that, okay, we're actually going to see less eye contact in general when they're looking at the human partner versus, just the human partner, and they'd be kind of looking more at the background. I think one reason could be because we were using participants who are higher functioning, so they might be a bit better at like adapting to these type tasks like this. But it could also be because of the smart glass, because the smart glass actually makes it so that it forces you to look at the eyes of the individual. So even if it's uncomfortable, if you're in this opaque glass and then all of a sudden it opens up, you are like forced to look at the eyes, versus if there wasn't a glass at all or something to kind of stop that, you could have been just wandering around. So I think because the task was so controlled, that's probably why we saw these differences, didn't see these differences.
Awesome. And yeah, also, I'm just going to squeeze in a question really quick, kind of in line with this. You mentioned this in the beginning, but autism spectrum disorder is so heterogeneous. I was wondering, do you think that I actually don't know very much about ASD, so please correct me if I say anything, inappropriate or wrong. Is there different types of ASD, and what types of ASD were your participants, and do you think that could have had also an impact?
Yeah, so I think this is definitely a problem in a lot of like research that includes like neuroimaging techniques such as FNIRS and fMRIs, that we tend to have participants who have an IQ of 70 or higher. And that's partly because of you have to give instructions, and also like for somebody to be standing still in the scanner, or even for FNIRS, yes, it can tolerate head movement, but not like abrupt head movement. Otherwise, you're going to lose a lot of the data. So we did have a lot of participants who were more on the higher functioning side. I think something that I do want to do in the future is try to find ways in which we can help include the populations who have intellectual disability, because those are the people who are probably going to be also the most like maybe not as functional in society on their own. So yeah.
Excellent. Thank you. You just have a very positive note from Marguerite Matthews. Great presentation. No notes. Top clap.
Thank you.
You have another question as well from Nick Johnston. Thank you so much to Dr. Parker for an amazing talk. Yes, agreed. I was wondering if she had any papers, et cetera, about Black exclusion from fMRI due to hair. That's super important. I helped coordinate a study that's involving fMRI and want to talk more to my bosses about this problem. The study has been tracking enrollment, but I don't think we have data on racial ethnic makeup of the people who have actually done the MRI portion of the study. And I think that was really important, something that I think a lot of people, especially considering, the whiteness in academia, that just has not been considered when developing these tools and techniques. So.
Yeah, so because I was focusing on this, I realized there wasn't a lot of studies that had been looking into this. So there's the Choi paper, Choi and colleagues, that was looking into EEG, looking at exclusion in Black and Brown individuals. The paper that we just published in January in Nature Neuroscience, so my colleague Jocelyn Ricard and I were first author. We were looking at MRI, FNIRS, and EEG. There actually isn't like quantifiable data on like how many participants are Black, and the reason they were excluded because of hair type. And it's probably because of the underreporting that I talked about, because we did this in FNIRS, and it's a paper that just got accepted by Jasmine Kwasa and colleagues. And we were looking at the demographic data of FNIRS, and we saw that not many people were reporting if it's because of hair type or skin type. I think if we can kind of think of what could be happening with MRI, is just the actual like in case that we have when you go inside the MRI board, like if you have braids or if you have a wig especially, because a wig has like metal tracks on it, you can't go into the MRI. So those, I guess, are kind of candid examples, but I think we definitely need to be able to actually quantify this in numbers in future papers.
And we are at time. You do have 2 more questions, which if you don't mind staying an additional 2 minutes, we can answer. But for those that do have to go, I really just want to say thank you so much for attending this phenomenal Black and Neuro seminar series. The recording will be on our website and YouTube channel at blackandneuro.com. Please also just go sign up for our newsletter, and we really do appreciate the time. Do you have time, Dr. Parker, to answer 2 more questions? Okay, great. So again, thank you for everyone who has attended so far. We really appreciate the questions. Okay, another question. Oh, Jaw Cook just said, great talk. You're so inspiring. Okay. Yes. A question from Jay Gamble. First, thanks for presenting. Second, do you think individuals who undergone ABA therapy would have an easier time with this task than others?
Yes, I do. There were a couple of participants who had done similar tasks before, and you could definitely tell based on like the comfortability that they were a bit better at kind of adjusting to something like that. So yes, I definitely agree that I think depending on the treatment, it could definitely make a difference. So I think it's probably really important for us to try to do this in children, like before they get a lot of like ABA therapy.
And for those who don't know, because I don't know, what is ABA therapy?
So yeah, so that's like talking about behavioral therapy. So you could have like a therapist. Yeah, yes, yeah. Mhm.
Got it. And then the last question from Eric Klobenton. I'm so sorry, I did not pronounce it right, but is there any eye gaze or joint social attention research on individuals who have suffered trauma to the dorsoparietal regions? Would we also expect to see changes in eye gaze behavior in these individuals as well?
That's a very good question.
Mhm.
So I think I am not 100% sure there has been research on that particular, since it seems like an area that you would think would probably be very, very important.
Mhm.
But I would hypothesize that if you did have some disruptions in that area, that there probably would. Oh, actually, there's actually something that we were doing in our lab a couple of months ago. So this is not necessarily like there was like an actual like lesion, but we were using TMS to try to kind of disrupt that pathway. And we were doing it in a pilot study a few months ago. And what we saw is that with a typically developed person, when you did the TMS stimulation, they saw more jittering of the eyes than when they didn't have the stimulation. So that is indicating that stimulating that area does kind of create this kind of like differences in type of eye movement and eye jitter. So it's actually pretty interesting. I'll definitely like keep that in mind, and if that ends up being a paper, I'll definitely like share that.
Amazing, thank you. Well, that's it. Nick Johnson said, thank you so much for your answer. And Danielle said, great presentation, Dr. Parker, sorry. And Eric as well said, that's really interesting. Thank you for your response and presentation. So I think that's it. Again, I want to echo how amazing a researcher I think you are, a person you are, and an advocator. Thank you so much for sharing your journey with the Black and Neuro community. And again, I'm just going to repeat what I said earlier. I really appreciate you sharing with us, and I really can't wait to see what you do. I did have a comment though. You quoted yourself. You got to add now the PhD after your name, or just put the doctor in front. So don't forget that part.
Yes.
All right. Thank you all,
Me.
everyone, so much, and we appreciate you again. Yeah.
Bye.
Well, thank you so much. I don't really know how to switch back to the panel thing. There's just a few people here, but we stopped the recording, and so I really